Hello friends, welcome you all in IIT PAL Biomolecules third session of class 11th NCERT Biology book. I BP Shah PGT Biology Kendriya Vidyalaya Kesapuram. Before going to start my actual lesson, you remember I have asked some question. I think all of you are able to answer these questions. My questions was, name 2 structural polysaccharides which found in fungi. Answer is cellulose and chitin. The second question was, arrange the following carbohydrate in the order of increasing complexity of chemical structure. Increasing complexity means, I have already told, in the form of molecular weight, that is triose, which is a monosaccharide having 3 carbon atom, example glyceraldehyde and dihydroxystone phosphate. Next one, fructose, that contain 6 carbon atom, and next one maltose. Maltose, it is a type of disaccharide which is made by glucose and linked with the help of glycosidic bond. And next one, on the basis of their molecular weight, oligosaccharide, that is trisaccharide, tetrasaccharide, pentasaccharide and so on. Oligosaccharide, I have gave one example, that is raffinose, which is made by glucose, galactose and fructose, and the last one which have high molecular weight, that is starch. So, in this sequence, these are the sequence, that is triose, fructose, maltose, oligosaccharide and starch on the basis of their increasing complexity. Now, I have discussed the classification of biomolecules, that is first of all carbohydrate, next one proteins, next the third one nucleic acids, lipids are there. So, in this session I will discuss about the proteins. You know, the term protein is derived from the Greek word, that is proteus, which means first rank. It means protein are the biomolecules which occupy first rank in our cellular life and the term was given by Berzelius in 1838. It play very significant role in the structure and metabolism of the cell. You know, the water molecule which is abundant amount, its abundant amount present in the protoplasm. Next to the water, the protein present in our cell, it about 12% of cell component. Next to the water. Protein are the biomolecule which is made by carbon, hydrogen, oxygen and nitrogen. This is very common, which are present in all types of protein. Beside these, there are sulfur and other molecules also, phosphorus also present in the molecules. Beside these, the protein molecule contains sulfur, phosphorus, iron and other elements. if protein have sulfur group, that is called sulfur containing amino acids. We have discussed later on. Proteins are generally linear unbranched polymer of amino acid. As the term indicate, each protein molecule are made by amino group and acid group, that is NH2 group which are basic in nature and COOH group that are acidic in nature. And this amino acid when form polymer, it combined with the help of peptide bond. These peptide bond are found in between one amino acid and next amino acid, and the peptide bond is formed. We will discuss later on. There are 20 different types of amino acids are there. This has been classified on different basis, that is essential amino acid, non-essential amino acid, acidic amino acid, basic amino acid and neutral amino acids. So, about 20 different type of amino acids are there, which have great role in our body function, in our metabolism. These 20 different amino acids firstly divided into essential amino acid and non-essential. It is highly complex structure, very large molecules, high molecular weight. Example of one protein, you will very well hemoglobin. It is the chief constituent of our blood. Its molecular weight about 68,000 Dalton and its molecular formula is C 3032, hydrogen 4816, oxygen 872, nitrogen 780, sulfur 8 and phosphorus iron 4. So, this is the molecular structure of molecular formula of the hemoglobin, and its molecular weight about 68,000. Besides this, there are large number of amino acids like albumin, which is made by 582 amino acids. Insulin, you already aware, it is formed by the beta cell of pancreas. It is made by 22 and 31 amino acids, and there are two type of polypeptide chain are present in insulin, that is alpha chain and beta chain. Now, the different types of amino acids which form the protein, the basic structure of amino acid you will find here. Basic structure of amino acids. As we are aware, the amino acid which contain acid group as well as amino group, acid and amino group. The basic skeleton of amino acids is the centrally placed carbon group. As we know, it is the acid group in one side, and in another group, another side NH2 group is there. Here hydrogen group, and this one group is variable. this one group, that is R1. Variation of this group, there are different type of amino acid, that is 20 different type of amino acid is formed. So, if the R1 is replaced by H, so the structure will be COOH, NH2, these are the common form, and if this R1 is replaced by H, this form glycine, and their 3 letter abbreviation is GLY. Next, if the R1 is substituted by CH3, that is methyl group, just see this is the basic form COH, here NH2, H is there. Now, if this here is R1, R1 is substituted by CH3. Now, this is the amino acid called alanine, and its abbreviation is ALA. If R1 is substituted by CH, methyl group here, methyl group here. Now, the basic skeleton is this COOH, here is NH2, here is H. This is the structure of amino acid called valine, its abbreviation is val. So, when we substitute the R1 with different alkyl group or different molecules, the different types of amino acid is formed. Now, see the another amino acid structure. If R1 is substituted by CH2OH, it means alcoholic group, then what happen? The OH, CH2, this one is the basic structure COOH, here H, here NH2. So, if this R1 is replaced by CH2OH, then the amino acid is called serine, and its abbreviation is SER. If the R is substituted by CH2 COOH, then what happen? The R is substituted by COOH CH2, then the you can write the basic form COOH NH2 and H. So, this is called aspartic acid. Aspartic acid, it is ASP. So, when you understand the basic skeleton of amino acid, you are able to write different types of amino acid. Only 20 type of amino acids are there. Here I have written 5 or 6 different type of amino acid, only change in the R group, and the 3 group of the carbon, that is equal in all cases. Now, only you understand the changing of R1 group, you can easily make different types of amino acids. So, here you can see in this aspartic acid there is 2 COH group, COH here, here COH group. It means in aspartic acid there is the dominancy of acid group. So, this type of amino acid, when there is large number of COH group are there in comparison to the amino group, that type of amino acid are considered as acidic type of amino acid. So, we can say the aspartic acid, as the name indicate, acidic amino acid, acidic amino acids. So, you can easily write the acidic amino acid. In acidic amino acid, the number of carboxyl group much more in comparison to the amino group, that is the basic group. When the carboxyl group is much more, the nature of amino acid is acidic. When the amino group is much more, the nature of amino acid is basic. When the number is equal in both case amino group as well as acid group, that type of amino acid are known as neutral amino acid. Here you can see in that cases what happened, that there is only COH group and amino group are there, one amino group and one acid group are there. So, there is the balance, amino group and acid group is 1:1. So, serine you can consider, you can tell the serine is a type of neutral amino acid, neutral amino acids. So, you can easily understand about the acidic, basic and neutral amino acid. Let us write some another amino acid which is very important, that is cysteine. Generally amino acid I have already discussed in our definition, the amino acid which contain carbon, hydrogen, oxygen, nitrogen, these are the basic. Besides this, the sulfur containing amino acids are there. This sulfur amino acid containing amino acids are methionine and cysteine. When you write the structure of methionine, same NH2, here carbon, here COH group is there, now here is H. Now, the R1 R is changes into CH2, here is sulfur. If R1 is substituted by CH2SH, then the structure will be, the structure of amino acid is called cysteine, and its abbreviation is CYS. Now, the another type of sulfur containing amino acid, that is methionine. That abbreviation is MET, methionine. Methionine is known as, it is the starting signal of protein synthesis. When that translation is going to be start, methionine acts as a initiator codon. So, methionine is very important amino acid in class 12th when you learn the chapter protein synthesis, that is 2 part transcription and translation. In translation, when translation elongation of initiation of polypeptide chain is going to be start, this methionine acts as a initiator codon. So, you must know about the structure of methionine. The structure of methionine same in 3 side, no problem, that is COH group, here NH2 group are there, here H group are there, but here is the variation, that is CH2, sulfur and CH3 methyl group are there. So, when you understand this variation, when this variation are CH2SH, the structure is cysteine. When this variation is CH2, CH2S and CH3, this structure is called methionine. So, now I hope you all will be able to understand the basic structure of amino acid and different types of amino acid, how the change of the alkyl group, that is the R group, changes the different types of amino acids. Okay. So, these are the various types of amino acids are there. These amino acid form in our body, most of them are formed inside our body and many of them are supplemented through the food. Now, the amino acids, how the amino acid going to be combined? When the I have already discussed protein is a polymer of amino acid. When more than one amino acid going to be combined, it form the peptide bond. These peptide bond are formed by the removal of water group between one amino acid and the another amino acid. In between two amino acid, the water molecule is going to be removed and there is the formation of peptide bond. How the peptide bond is formed? Here you can easily understand. Take two amino acids, that is I have already discussed. One, you have take the glycine. Glycine is the simplest form. This is the COOH. Here is the NH2 group is there. Here is H. Here is H. The R1 is H. This is the structure of glycine. You may consider glycine as amino acid 1. Now, the next amino acid you can write here, that is the next to the glycine simpler one, that is alanine. This alanine are only change, the CH2 group is replaced by CH3 group. So, we can write here NH2 as this one, here H, here COOH, here CH3. Now, you are able to write the structure of alanine. Now, you can treat the alanine as amino acid 2, this one amino acid 1. Now, the amino acid 1 combined with the amino acid 2, how it is going to be combined? The amino acid, first amino acid OH group and second one amino acids H group is going to be combined. After the combination, it remove the water. This water is going to removed and the bond is formed. These bond are of this type H N. So, this bond now after the hydrolysis removal of water, this bond is called peptide bond. Okay. Now, we can write this structure in the similar way and name this. So, we can easily write now this one H, CO, H, NH2, here is HN, C, here is CH3 methyl group, here is COOH group, here is H. So, this one is glycine. So, we can abbreviate it GLYCYL. Here is alanine, ANINE, glycine. So, now the name of this protein is glycylalanine. Now, the peptide bond are here. So, now you can easily understand how the peptide bond is formed in between the two amino acid and water molecule is going to be removed and the peptide bond is formed. Okay. Now, by the combination of different type of amino acid, different types of protein molecule is formed. This protein molecule when is going to be synthesized, so we can easily say protein are the polymer of amino acids. It is most abundant biomolecules of the living system. Chief sources of proteins are milk, cheese, pulses, peanut, fish, meat. They occurs in every part of our body and form the fundamental basis of structure and the functions of life. They are also required for the growth and maintenance of the body. So, it is called building block element of our body. Amino acid, as we have already discussed, in alpha amino acid the amino group located in the alpha carbon. Now, classification of amino acid, just I have already discussed. The amino acid which can be synthesized in our body are known as non-essential amino acids. Example, glycine, glutamic acid. Here the term is acids are there. On the other hand, those which cannot synthesize by our body, it can be supplemented through the food, which cannot be synthesized in the body and must be obtained through the diet or through the food, are known as essential amino acid. So, essential amino acid are those which can be not synthesized by our body, it can be supplemented through the food. There are 8 different type of amino acids, essential amino acids are there. Essential amino acids are of 8 type. So, we can understand the total number of amino acids is 20, where the 8 are essential. So, the non-essential amino acid 20 minus 8, that is 12. 12 different types of non-essential amino acids are there, which can be synthesized by our body. These 12 can be synthesized by our body, these 8 not synthesized by our body. The name of these essential amino acids are valine, isoleucine, phenylalanine, methionine, leucine, lysine, threonine, histidine, etc. So, these are the essential amino acid which are not synthesized by our body. Rest one will be the non-essential amino acids. Now, amino acids, as I have already discussed, has been classified on the basis of presence of amino group and the acid group in that particular amino acids, acidic, basic and neutral depending upon the relative number of amino and carboxyl group in their molecule. If equal number of amino acids and carboxyl group make it neutral, it means the number of COH group and number of amino group will be same, that is 1 by 1 ratio, that is called glycine. More number of amino acid, it means when the basic character of the amino acid is much more amino group than the carboxyl group makes it basic. Example of basic amino acid lysine, when the more carboxyl group are there, it means the acidic character of amino acid becomes high. So, that type of amino acid are called acidic amino acid, like aspartic acid as we have already discussed and glutamic acid. These are the different types of amino acid on the basis of their relative number, which are present in the different type of amino acids, amino group and the acid group. Now, there are the different types of proteins on the basis of when the amino acid is going to be synthesized, it constituted the proteins. These proteins, it is a heteropolymer heteropolymers containing string of amino acids. Proteins are formed by the amino acid, no doubt, with the help of peptide linkage. It is a polymer of alpha amino acids linked by peptide linkage. As we have already seen, these peptide bond are formed in between COOH group and amino group. If proteins are made by only 2 amino acids, that protein is called dipeptide, because in the formation of dipeptide only 2 amino acids are involved. Now, after the formation of different types of amino acid and linked with the peptide linkage or the peptide bond, different types of protein on the basis of their structure is formed. That protein has been divided into structure of proteins. Structure of proteins. On the basis of their structure, the protein has been divided into primary structure. Next one, secondary structure. Next one, tertiary structure, and last one, quaternary structure. On the basis of arrangement of amino acid, how the amino acids are arranged at the time of formation of peptide bond, these 4 types of proteins are there. When in primary structure, of protein, there is linear sequence of amino acid. Linear sequence of amino acids, that means which form polypeptide chain. Polypeptide chain, and the sequence are of this type. Suppose one amino acid here, the second amino acid here, third amino acid here, fourth amino acid here, fifth amino acid, and so on. When these are the sequence, or you may make this type, this is called primary structure of protein. And this one bond, this is the amino acid 1, you can write amino acid 1, 2, 3, 4. In between all the amino acids, there will be the peptide bond. If at the time of protein synthesis, if the amino acids are linearly arranged and the peptide bond in between them are present, then this sequence is called primary structure of protein. Next one, secondary structure. Secondary structure of protein. When the amino acid are formed and combined with the peptide bond, it form a coil like structure, just like this. You may write here, here is the one amino acids, here is the one amino acid, this form coil like structure, and in between them there is the peptide bonds are there. And after the coiling, it may make 3-dimensional structure. So, we can say the polypeptide chain is coiled to form the 3-dimensional structure, all called secondary structure of protein. Now, the third one type of protein are tertiary structure. The tertiary structure of protein, it is the helical polypeptide, which further coiled and again folded and folded to form complex structure. It forms the complex structure, that is called tertiary structure form. So, we can see the structure of tertiary structure, it is going to be coiled, further coiled. So, this type of structure of amino acid, and in between them peptide bonds are there. So, this type of structure is called tertiary structure of protein, which is found in the folded form. Now, the last one structure of protein, that is quaternary. Quaternary structure of protein. When the amino acid are formed and it form polypeptide chain, this polypeptide chain assemble to form various types of polypeptide, alpha polypeptide chain, beta polypeptide chain, and several types of polypeptide chain. When becomes a complex structure, it forms the quaternary structure. It means the more than one polypeptide chain assemble to form quaternary structure. Just like when you see, this is the one type of protein, and in that cases the another types of helix are also assembled. This is called, suppose this one is the alpha form, this one is the beta form are there, all is going to be combined, all assemble. Here is the alpha chain and beta chain combined to form the complex structure. This is called quaternary structure of protein. So, there are 4 types of protein structure on the basis of their coiling, or if the coiling is very simple like linear fashion, that structure is called primary structure. If it is going to be coiled, simple coiling are there, then form the secondary structure of protein. If the further coiling is formed, coiled, and that becomes the folded form, that is called tertiary structure of protein. If more than one polypeptide chain assemble, then it form the quaternary structure of protein. The example of quaternary protein structure of protein, that is hemoglobin. This hemoglobin, we have just seen the molecular weight and their formula of hemoglobin. The hemoglobin are made by different types of peptide chains, and the structure are of quaternary structure. Now, after the protein, there is lipid. Lipid is also a type of biomolecules which molecular weight is very high. Lipids. it is also called heterogeneous group. Heterogeneous group of biomolecules. It lipid include fats, oils, waxes and other materials and other substance. Generally, lipid are very important molecule because it is the chief constituent of the plasma membrane and its very important role in the enzyme formation as well as in carrier compound. So, lipids are generally, it is the characteristic feature of lipids, it is generally insoluble in water, but considerably soluble in organic acid. This organic acid may be benzene, may be chloroform, may be ether. So, in the generally lipids are insoluble in water, but soluble in organic solvent like ether, benzene, chloroform and etc. These are hydrophobic in nature. These are hydrophobic in nature. Now, we can classify the different types of lipid. Generally, the lipids, the main constituent of lipids just like carbohydrate, carbon, hydrogen, oxygen, but in carbohydrate the carbon, hydrogen, oxygen are present in the fixed ratio like Cn, H2n, On. These are present in carbohydrate as we have already discussed. But in case of lipid, these three atoms carbon, hydrogen, oxygen is also present, but oxygen is very less amount. So, for the oxidation of lipid it requires much more oxygen. So, after the breakdown of lipids it gives much more energy in comparison to the carbohydrate as we have discussed in my chapter respiration. The respiratory fuel lipid gives 1 gram of lipid, it gives 9.2 kilocalorie of energy, while 1 gram glucose when oxidized it gives 4.1 kilocalories of energy. So, it is because lipid contain less oxygen in comparison to the carbohydrate like glucose. So, for the oxidation it requires much amount of oxygen. So, lipid gives more energy in comparison to the glucose molecule or any other type of carbohydrate and the protein. Now, classification of lipid. Classification of lipids. Lipids can be classified in major 3 groups. One is simple, that is called simple lipid. Second one conjugated, and third one which is formed from the derivation of simple and conjugated form, that is called dried form of lipid, that is dried form of lipid. So, we can classify lipid into simple, conjugated and dried form. Simple lipid, it has been also divided into 2 forms. This one is called, first one is called true fats, or it is also called natural fat or oils. This natural fat or oils, it is simply formed by the combination of fatty acid and glycerol. everyone, can you understand, when the acid combined with the alcohol, it form the bond. Which type of bond it form? Okay, very right. This is called ester bond. So, with the help of ester bond, the fatty acid combined with the glycerol molecule, combined with the alcohol molecule and form ester. The simple lipids, simple fat, it is made by fatty acid and glycerol. The one glycerol molecule we have already discussed in the carbohydrate, in glyceraldehyde, glycerol it contain 3 OH group. So, for the combination of 3 COH group, it required 3 fatty acid. So, simple fat are made by 3 fatty acid and 1 glycerol. So, 3 molecule of fatty acid when combined with the 1 molecule of glycerol, it form a simple fat that is called triglyceride. In the presence of enzyme that is called lipase, 3 water molecule is going to be removed and the bond form that is called ester bond. Now, the true fat, the second one is waxes. Waxes when the fatty acid combined with other than alcohol, not glycerol. So, we can simply understand, if the fatty acid combined with the glycerol, that form true natural fat. If other than glycerol, other alcohol is going to be combined with the fatty acid, this form waxes. Okay. Fatty acid and other alcohol, it form the waxes. Now, the conjugated form of the glycerol, this form, no doubt the conjugated form of lipid, it is formed by fatty acid, no doubt, fatty acid plus alcohol. Beside this, the other substance are also there, because without fatty acid, without alcohol, without glycerol, there is no lipid formed. Fatty acid, glycerol are commonly present in the lipid. Beside the fatty acid, glycerol or the other alcohol, that the other molecules, other atoms are also found that form the conjugated form of the lipids. These conjugated form are divided into 3 main types. These 3 types are like phospholipid, as the name indicate, lipid, fatty acid and alcohol are present. After the fatty acid and alcohol, there will be the phosphate. It means fatty acid, alcohol plus phosphate. These form the phospholipid, which is the main constituent of the plasma membrane. The second one, conjugated lipids are glycolipid. Glycolipids, as the term indicate, there is no doubt it is formed by fatty acid plus alcohol plus glycon. Glycon is a type of carbohydrate. If fatty acid, alcohol combined with carbohydrate, that form glycolipids. The third one form is lipoprotein. As it indicate, the lipoprotein are for phospholipids, which is formed by fatty acid, alcohol and phosphate. Beside these 3 things, there is the addition of protein. So, lipoprotein are made by phospholipids, that means fatty acid, alcohol, phosphate. Beside this, protein molecules are there. So, 3 types of conjugated lipids are there, that is phospholipid, glycolipid and lipoproteins, and you can easily understand this how these 3 things are constituted beside the fatty acid and alcohol. Now, the third one form is derived form. As the name indicate, it is the derivation form, derivative form of simple and conjugated. So, we can easily define as it is formed during hydrolysis of simple and conjugated, simple or conjugated lipids. The best example of derived form of lipid in your book, that is steroids, sterols. So, these are the 3 basic form of lipids, that is simple, conjugated and derived. Now, the how these lipids are formed from the fatty acid and glycerol. Simply we can define the fats are the ester of glycerol and fatty acid, that means that triglyceride. How these triglycerides are formed and the ester bond is formed. You can see here at the time of formation of ester bond, we need the glycerol molecule which contain 3 carbon, that is OH, here HOH, here 2OH. This is the structure of glycerol. Glycerol 1 mole. 1 mole of glycerol, it has 3 OH group. When it combined with any type of fatty acid like palmitic acid, its formula is OOC C15 H31. When 3 molecules of palmitic acid combine, OOC C15 H31, C15 H31. When 3 molecules of palmitic acid, 3 moles palmitic acid combined with 1 mole of glycerol, it form here 1 hydrogen combined with the OH group, it release 3 water molecule. After the releasing of water molecule, this is going to be combined and form triglyceride. And this reaction occurs with the help of enzyme lipase. So, with the help of lipase enzyme, the 1 mole of glycerol combined with 3 mole of palmitic acid, that form after the removal of this structure is formed, because water is going to be removed. Now, this one bond is called ester bond. So, you can easily form the fatty structure, that is the simple fat is called triglyceride, because the triglyceride required 3 molecules of fatty acids and 1 molecule of glycerol. So, there are 2 type of fatty acids on the basis of unsaturated and saturated. If the carbon in between the carbon there is the double bond or triple bonds are there, that is called unsaturated fatty acid. In between carbon and carbon there is single hydrogen bonds are there, that is called saturated fat. Saturated fat have high melting point, boiling point. So, now you are able to understand all these type of questions related to the lipids. Now, think about these questions. How many types of amino acids are found which occurs in protein? Name any 2 types of aromatic amino acids. Name the nitrogenous base which are present only in the RNA. Name the chemical constituent of the nucleotides. Name the sugar which are present in the nucleic acid. This question will be solved in my next session of biomolecules. Thank you.